Okay, I have uploaded several things to the OneDrive. Several of you asked me to upload the PowerPoints. Those are now available week 1 and week 2 under my name. If you go to the Biology 3311 Genetics Lab, I have renamed mine to Ricky's Files. So I'll put files here. I'll continue to put the videos on YouTube. So you have the PowerPoints, you can download those, look at them. I also have your assigned mutants in case you lose your copy that has your assignment on it. You can go get it again here. The thing that I think will be of most interest to you will be this. I have uploaded a writing assignment 5 from a previous semester. So let's take a look at that. I'm going to open this up here a second, and this video will be on YouTube, but you'll have to come to get the file, you'll have to go to the OneDrive. So let's open this, and here we go. Now, this assignment is not the same as yours. The assignment you will be given will be similar, but not the same, but a lot of the same requirements are going to be made in your assignment that are made here, and it's always good to just see an example. So the content will be a little different because you aren't working in the lab and raising flies. Instead, you're going to be doing more bioinformatic analysis. So realize here where they're often talking about crossing flies and what it was like in the laboratory, you're going to be maybe looking at sequences. And so realize the content will be a little different, but the format for the paper will be pretty much the same. It's not set in stone yet, but understand, I just thought it would help you seeing this paper, seeing what the expectations are, and the expectations are quite high. You may or may not be able to write a paper as good as this one. This was an A+. Don't let it intimidate you in that sense. If you're not a good writer, use this as a guideline to become a better writer. So we'll start with just your first opening. It's writing assignment 5. All right, and then this will be I don't know if the actual opening page like this is an actual requirement, but certainly this page is. And this page, let me zoom in a second. I'm pretty sure that you're going to have to do something very similar to this for the paper. Now the name will of course probably be different, and we're not doing so much of the classical and molecular genetics as we are bioinformatics, so you might have to change the name a little. Probably a good idea. You'll be doing more of a bioinformatic analysis, sequence analysis, terms like that can be used. But the idea is you will be analyzing a gene and you will be using Drosophila. So keep that in mind. You'll need to do something like this. Get the dates right, of course. Put your names in there. You don't have group members, so it's not an issue. So a lot of this you won't have to do. Name and ID, and of course you won't have things like this over here. But the first thing that comes in a paper like this is an abstract, which is like a summary, kind of getting everybody oriented on what you're trying to accomplish in this paper. So notice that that is roughly a page. Okay, that will be the first thing that you have to put into the paper. Now you're going to be combining all of your WAs, the content in those, into this large one. So by knowing what your large one looks like, it might help you get a feel for what you need to do for WA1, 2, and 3, and 4. I'm going to try and find some WA1, 2s, 3s, and 4s to help you out. But one thing I did find very easily, I had a WA5 here ready to go for you. So I'm going to show you this with the idea so that you can have an idea of what you're up against. Oh, that sounds bad. The sense what you're up against is, yeah, it's a long paper. It's not going to be easy. maybe this is some good incentive to get started early. Notice here that the italicized and the capital D here for Drosophila melanogaster. All right, or melanogaster, depends if you're American or European. So capital D, small m, italicized for the name of the fly. Now you'll notice that later on when you do this, but then you get to switch. The abstract is separate from the paper, so you have to have the full name in the abstract. But in the paper, you do it once, and then you get to do D dot melanogaster. All right. Notice the references and how you reference the papers that you will use to support your ideas. All right, here. Note that there are figures that you will create and tables that you have to reference. If you put the table in the back of the paper, you don't put them here directly. You put them in the back of the paper and you reference them in the middle of your paper in the content area, and they will be located at the end of the paper, and I'll show you those. So keep all of this in mind. You do have to get all the formatting right. You know, the right margins here, the right font, all of this. Okay, so make sure you get all of that. Now notice when you need to italicize, capitalize, proteins, alleles, genes, name of organisms, all of those have to have the right format for you to get full credit. All right, you'll be taken off if you miss it once or twice. I might not notice, but if you're consistently getting it wrong, you'll definitely be taken off, and it's not a minor amount. If I'm not a stickler on it, but look, if you do it over and over again, I'm going to take off, and I'm going to take off probably a lot. There is a set amount I'm supposed to take off for you not getting the format right, and if it's something that you should have known, like italicizing something, it's going to cost you. Here's a good one. I like that this person recognized that, you know, you're at the beginning of a paragraph and you don't capitalize it because she started with an italicized name there, and it's not supposed to be capitalized, and this person did not, and that is the correct format right there. So understand that you need to know the rules for doing this. Now once we get through the introduction, which the introduction will probably be quite similar between one of these papers and ours. We're going to be doing more bioinformatics, and that will alter the materials and methods more than anything else, because you're not going to be able to go into the lab. You're going to be doing data analysis essentially. So recognize that you need to go through here, and you need to, as you read this, realize fly strains and how crosses were conducted. You will be doing this, but it will be virtual. It will not be in the lab. So a lot of times in this, you'll have to alter things to compensate for the fact that we're not online. That's why this paper will be different than yours. The structure is generally the same, which will help you out quite a bit. You'll get to see the structure. You'll get to see how to write a paper here, but your content will be different. You'll have to change like how phenotypes were scored. You're not going to be doing this at all. This is what you do when you have flies that you're looking at, and you kind of have a set of rules for determining whether or not, say, this fly had curly wings or not. And you're going to have to figure out, or when you do it this way, you have to figure out, okay, is this really a curly wing fly or is it not? How do we actually set this up? What did we find? That sort of thing. Scoring. All right. Now, you're not going to be able to do that. You're going to have software that's just going to be doing virtual flies. There's no need for scoring. However, you'll be doing data analysis. You might have some tool here for like maybe something called a dot plot or something that you're going to be comparing sequences with, and maybe you would discuss that here. There will be statistics. This part will not change much, so make for sure that you take a look at this and understand what they're saying here. When you do, then you know you're ready to start writing this part. If you can read this and understand what they talk about, then you're in good shape. So I would definitely, when we get to chi-squared and some of the statistics, come back here maybe and read this. And when you got it down enough to understand what's going on here, well, maybe it's time for you to start writing this part. We will not be doing the DNA extraction. Now, we might be asking you to explain this in some manner. Now, you didn't do the actual experiment, but maybe we will ask you how you would have, or if you had obtained something like a particular result, what that would have meant. If maybe we'll give you fake results and you'll have to tell us what they mean. I don't know how we're going to work that yet, but just keep that in mind. These are the things that we did when the class was in person. Of course, we can't do these things in person right now, so we have added more bioinformatics. So you'll be, instead of doing this type of work, you'll be doing more data analysis, which is what bioinformatics entails. All right, so here is BLAST. BLAST is a bioinformatics tool, and they did do BLAST in the previous class, so you will certainly be doing it here. So here's an example. So when we learn about BLAST, you need to come back maybe here and read this and make sure you understood what they were saying. And if you do, then again, maybe that means it's time for you to start trying to write some of this. Finally, you end up with your results. Actually, there'll be a discussion after this as well, but the results are what you found out. Now, their results involved their crosses. Your results will probably be some type of, will be crosses, but they'll be virtual. But your results will probably include a great deal of sequence analysis, of data analysis as well that we'll be using. Maybe it will be where you find the transcription start site of a gene, something like that. So those will be the results that you gain here, not necessarily crosses, though I suspect the crosses, we'll be doing them virtually. You'll have to include the crosses as well, of course. I would definitely read through this paper. This is an excellent paper, and a good mark for you to work from. A good example. Let's scroll down. Yes. Much of this you will not be doing. So the molecular basis of PCR, I'm certain we need to teach you about PCR and gels. We won't be able to do them, but you're going to at least need to know how they work. So I would think that there needs to be some of this in your paper. Maybe not, might not be emphasized, but certainly that you understand how to interpret them. In addition to this, you'll probably have to also do some type of bioinformatic analysis and have some graphs or tables dealing with something more data-driven. All right. More BLAST, like I said, BLAST is a bioinformatic analysis, so certainly this would be part of your paper. Discussion, this is where you kind of go through and kind of bring it all together and say, hey, we found this is what we think happened, this is why we think as we do, what kind of results did we get that support our hypothesis that something is true or not true. And this is where you would discuss this. All right, and this is a quite lengthy part, and we should be getting close to 20 pages written. 20 pages written. That's I suspect that'll probably be what you were asked to do, something in that range, maybe a little longer, a little shorter. Now, that's just the written part. There are also references. All right, this person had 10 or 12, 11. They had 11 references. That's a little maybe on the short side. I don't know. About right, I guess. Um, but you do have to, of course, reference those in the text. Here are the list of the references, but you had to, cite them within the text as you use those. So keep that in mind, that all of your references must be cited in the text. Then comes the tables and figures. And the tables and figures can be as long as your paper. You'll have tables, figures, and you'll be probably more in if we're doing bioinformatics, where because normally you have pictures of the flies that you crossed in the traditional paper like this one. Well, you're going to have to be analyzing something closer to data, and you'll have sequences, and you'll probably have to display that. So there'll be more, when we get down a little bit, they do a little bit of the BLAST and some other sequence analysis. You'll probably be doing more of that, and so there'll be more figures dealing with those types of things than there are like pictures of flies and things like that. So let's take a look at some of these tables and figures. I'm just going to scroll through and not talk unless there's a need to. You will probably still be doing all of these things. So these tables are probably very similar to what you will be doing. You will more than likely need to do some type of cross explanation using these chromosome models. That's probably one that you will also have to do. Now remember, all of these tables need to be referenced in your written paper too. So you create these, and you're going to have to talk about them in your written paper, or you will be counted off. You certainly will be doing chi-squared, so this is a legitimate table here. You'll probably need to do something, as close to this as possible. Here's something you may not have to do much of. Now, these people took these pictures themselves, and they're pretty good. Some are better than others. A few are a little blurry. Depends on what you're trying to look at here. But you probably won't be doing as much of this because you don't have live flies to take pictures of. Now, notice you do have descriptions of everything. So you have to not only have the correct format down here for your description, but you need a description, and you need to reference the things up here, and then of course the figure itself must be referenced in the written paper. So keep all of that in mind as you move forward. Now here's the gel that was referenced earlier in the now we can't do that. Maybe we'll give you a gel and ask you, hey, what does this mean? And perhaps you would put it in the paper. I don't think we have that many pictures of gels though, but if we do, maybe somebody will decide we need to do that. I don't know, but I know that you will need to know how to interpret a gel by the end of the course, if you don't already know how to do that. All right, and the description, of course, and how that was made. Now here is more of the bioinformatics side. You'll be doing things closer to this. They had to do a little bit of this in the previous courses. You're going to be doing a lot of this. All right, but you reference the figure just like you would any other table or picture. You need to reference these as well. All right, description, name, figure 10. All right, and that's it. So this ended up being 37 pages. It was roughly 20 pages of written text and 17 pages of references and figures and tables. So they're about, it's roughly 50-50 written and tables and figures and references. So keep that in mind that a lot of this is in the tables and figures. Your length may differ in this class depending on how we do the bioinformatics and put this together. Keep in mind that the there will be fewer, of course, pictures like of the flies and more of this bioinformatics type sequence analysis. You might be finding a transcription start site or something. You may be finding splice sites in your gene, things like that. And if that's the case, you'll have to reference them with different types of figures. All right, I thought this would help. Actually, I'm sure this will help. I'm going to give you also a little video on Flybase and how to use Flybase, along with, let's see, what else am I going to give you? Maybe a little rundown of WA1 requirements. But this is WA5. It's to give you the big picture, kind of to show you where you're headed.